Good morning, everyone. I would like to welcome you to the fifth webinar of the Neuroscience webinar series, jointly organized by IATRIS and Neuroatris. Today, we are very pleased to have two lecturers from Neuroatris and France Life Imaging speaking about ultra-high field MRI and MRS for clinical and translational research. My name is Laurent Duquesne. I am project manager at Neuroatris, and I will be hosting this webinar today. I would like to make you aware that this webinar is being recorded, including the questions and answers at the end of this session. It will be made available on IATRIS and Neuroatris websites shortly after this session. We have reserved some time at the end of this session for questions. We invite you to write down your questions in the question box in the control panel, as you can see on this slide. Before I introduce our speakers, I will briefly present the three research infrastructures bringing you this webinar today. IATRIS is the European infrastructure for translational medicine. IATRIS helps you de-risk and add value to your drug, vaccine, or diagnostic development program. We do this by providing fast, tailored access to cutting-edge enabling technologies in translational research. Via our central hub, you can access the vast array of clinical expertise and high-end facilities that are available within the more than 80 top-tier academic centers across Europe. Always keeping the patient at the heart of our interest, IATRIS relies on three pillars to help early decision-making: biological insight, validated analytical tools, and clinical observation. IATRIS' strategic research agenda follows two main directions. From the technical perspective, we help select and validate next-generation translational tools and biology. From the operational perspective, we foster new collaboration and encourage new funding models. You can find out a lot more on our website, iatris.eu. Neuroatris is the French node of the European infrastructure IATRIS, and it is the only node whose specialization is solely neuroscience. Neuroatris is also a national research infrastructure dedicated to neuroscience, and more precisely the neurodegenerative diseases that are Alzheimer's, Parkinson's, Huntington's diseases, and multiple sclerosis. Neuroatris is composed of expert teams from 5 renowned hospitals and research centers accessible through a one-stop shop. This consortium brings together all the needed bricks to build a full translational neuroscience project and gives you an easy access to multidisciplinary expertise and high-level platforms to achieve your project in neuroscience from bench to bedside. All this information and more can be found on our website. Should you have any question about Neuroatris, please feel free to contact me or our business development manager, Ali. We will be happy to answer them or to put you in contact with the experts you need to define your project. France Life Imaging, or FLI, is a French national research infrastructure dedicated to biomedical imaging. FLI is a large-scale and distributed network of research platforms gathering 25 imaging facilities connected to research teams with a well-known scientific expertise in biomedical imaging. As such, FLI provides a convenient access to a multimodal imaging offering, including both imaging systems and integrated services. The facilities are organized into 6 nodes covering the French territory. In addition, a national node brings together French laboratories well known in information analysis and management. Research efforts are structured through 4 work packages, namely molecular imaging agents, instrumentation and innovative technologies, interventional imaging, and image processing. Finally, 2 national-level strategic work packages, one for training and one for management, complement the organization of the FLI network. For more information, please visit FLI's website, francelifeimaging.fr, or contact FLI by email. Our first speaker today will be Dr. Cyril Poupon. Cyril Poupon was hired by the CEA in 2002 as an MR physicist and moved to NeuroSpin in 2007, where he successively held the position of MR physicist in the laboratory of MRI and Spectroscopy and the position of expert in image processing in the laboratory of image analysis. In 2010, Cyril Poupon took the position of Director of the Laboratory of MRI and Spectroscopy of NeuroSpin, co-leading the two following transverse research programs: Pushing the Limits of MRI, targeting the development with his team of ultra-high field imaging methods, and Multiscale Human Brain Architecture, modeling the human brain at various scales, and developing methods to map the human brain structural connectome and tissue microstructure. Since 2017, he has become the Deputy Director of NeuroSpin, in charge of the MRI platform. Since 2018, Cyril Poupon is also chairman of IATRIS Imaging and Traces platforms. Our second speaker will be Dr. Fawzi Boumezbeur. After his PhD in Orsay, France, and 2 years as a postdoctoral fellow at Yale University studying normal and pathological energy metabolism and neurotransmission in the brain using in vivo hydrogen-1 and carbon-13 MRS, he became a physicist at NeuroSpin at CEA Paris Saclay in France. He specializes in magnetic resonance spectroscopy and non-proton magnetic resonance imaging at ultra-high magnetic field. His research is focused on developing, validating, and applying new MR biomarkers to investigate brain physiology, metabolism, and pharmacology from animal models to patient cohorts. I will now hand over the presentation to Cyril.
So let me introduce you why it is interesting to use ultra-high-field MRI and MRS for clinical and translational research. And to try to convince you, I will do a focus on neuroimaging, because as I mentioned, we are part of the NeuroTris node of IATRIS, and we are specifically doing neurosciences. So first, why to use ultra and high magnetic fields? And first, you have to know that, the nuclear magnetization is linearly dependent on the magnetic field, such that the strongest the magnetic field, the more signal you get in terms. So it will allow you to increase, the resolution you can have about many aspects of brain functioning and structures. For instance, you'll be able to better look at neurons at work, doing functional imaging. You'll be able to better look at the connections between the neurons, and you will be able also to better probe the metabolism of the brain, that will be explained later by Fawzi Boumezbeur. And it is of utmost interest to get access to such structural and functional aspects of the brain in order to better study brain disorders. And you can see here on this first slide a kind of example of imaging performed in vivo at 70 on a human brain, where you can already see some very nice details of the brain. And on the right picture of this slide, you can see what can be achieved post-mortem, where you can even go higher with a higher resolution to, for instance, here look at the lamina structure of the cortical ribbon. And so the quest of ultra-high field is just to improve the resolution, and we typically separate what we consider as high field up to 3 Tesla, ultra-high field between 7 and 9 Tesla, and extreme high field, which goes above 10 Tesla. And actually, the panel is that most of the clinical hospitals are equipped with 1.5T and 3T. But the democratization of ultra-high field system at 7 Tesla, that now has got their FDA approval and CE label for Europe, turns the use of ultra-high field to be almost clinically feasible, like you are doing imaging on a 3 Tesla system. So I will give you some example of what can be achieved, and I will first try to see that using ultra-high field first allows you to improve your spatial resolution of your data and to read the mesoscopic scale. So if I consider, for instance, one important structure, the hippocampus, which is the center of memory in the brain, that is represented here in red, then if you are using conventional MRI at 3T, you can see that the resolution of the image around your hippocampus is not so exceptional. And roughly speaking, you can get a resolution of 1 millimeter. But if you're now using a 7 Tesla MRI system, you can enhance that resolution up to 300 micrometer. And while you were not able to see the circumvolutions of the hippocampus, you can clearly see them at 7 Tesla. So you can clearly see that you enhance the understanding of the structures of your human brain. And now the target of ultra-high field is to do that at an even here field, and we call it extreme high field at 11.7 Tesla, and that will be the kind of data. So here, this data was done on a hippocampus sample on a preclinical system at 11.7 Tesla, and you can clearly see the nice structure of your hippocampus, and that is of utmost importance to have access to that kind of data. Because, I mean, when you consider the decrease of the volume of the hippocampus you observe in Alzheimer's disease, then you'll be able to investigate which subpart of the hippocampus is atrophiated or which connections between those subparts are abnormal. And you can see here a delineation of the hippocampus on the right side of the slide, where you can clearly see that you're able to segment CA1, CA2, and CA3 parts of the hippocampus. And if you're now doing postmortem imaging, you can even go to a higher resolution, close to 100 micrometer. Here, this is a kind of imaging that was performed in the frame of the Human Brain Project, a European flagship, where you can see that you can get access to histological-like MRI data sets. And you can clearly see here on the anatomy that you can distinguish, for instance, the fine vasculature, microvasculature of your brain. You can even see the laminar structure of the cortex, but you can also probe the connections. And for instance, in that case, it was a slice acquired around the corpus callosum, a big white matter bundles connecting the two hemispheres of the brain. And it is the first time that at 11.70, you can clearly see the 19-degree sharp turns of the thalamic radiation that were something that were mentioned by neuroanatomists, but no one could evidence that it was actually turning at 90 degrees. And that was one of the results of the use of ultra-high field in combination with strong gradients. And so this can demonstrate that, I mean, using ultra-high field and extreme high field, you can get very high resolution structural data. But not only you can get access to enhanced resolution data, you can also observe novel contrast mechanisms. And here, this is an example where here we have focused on a study about Parkinsonian syndromes. We all know that for Parkinsonian syndrome, the brainstem is a very impaired structure, and more specifically the substantia nigra and the subthalamic nuclei. And so when you are doing standard imaging at 3 Tesla, which is shown here on the top left picture, you can see those substantia nigra and red nuclei structures. Whereas when you are acquiring at 7 Tesla, because those structures contains a lot of iron, you can take benefit from the susceptibility effect contrast that clearly allows you to delineate finally those structures. And when using image processing on those kind of data acquired at 7 Tesla, then you can, for instance, delineate some very fine brainstem structures of interest. And now that you're able to, for instance, segment all those brainstem structures, you can just look at, for instance, the connections of those brainstem structures. And here I would like to focus on the pedunculopontine nuclei, which is one of the new targets of interest in the surgery of Parkinson's disease. And if you're looking at those pedunculopontine nuclei, and you're looking the connections to those pedunculopontine nuclei to the functional areas over the cortical ribbon, and you can do that because you have acquired also the brain connection at 7 Tesla, you can here represent a matrix that gives you the number of fibers connecting each of these structures to each cortical area. And if you are doing that for two populations, the populations of patients suffering from progressive supranuclear palsy, a kind of Parkinsonian syndrome, and you compare that to a population of control subjects, which are all healthy, then you will observe that there is an abnormal connectivity of the pedocoilopontine nuclei to the motor cortex area BA6, which is related to the gait disorders they are actually facing, but you can also see some abnormal connectivity to the globus pallidum and to the thalamus. So because you are using those novel contrasts, you are now able to answer some questions of interest to study the Parkinsonian syndromes. And if you want even to go at a high resolution, you can acquire the same data set on an ex vivo post-mortem human piece, and you will be able to segment and create atlases of all the structures of the brainstem that you cannot achieve in vivo. And you will be able to establish atlases that can be used after to better monitor the surgery in new patients acquired at 70, so at a ultra high field and not at extreme high field. And this is a kind of data that shows you both the interest of the enhancements of the resolution and of the contrast that you can achieve at 7 Tesla. And of course, ultra high field are compatible with translational project because you can use ultra high field from rodents to primates. So here on the left, you can see a kind of study that were conducted in the frame of a RHU project in France, where we were able to establish the myelin weight fraction maps of mouse in CADASIL syndrome. The CADASIL syndrome is a kind of degenerescence of the microvasculature that conduct to dementia. And some biological hypotheses tells that, I mean, the corpus callosum, which is still that big white matter bundle connecting the two hemispheres, has got abnormal myelin contents. So because you are using extreme high field at 11.7 Tesla, then you are able to reconstruct those data at 50 to 100 micrometer isotropic resolution, and then you can compare, I mean, your population of CADASIL model mouse with a control mouse. There are also some other examples. So you can, for instance, study other kind of animals like sheep and even some primates. So here, this is, for instance, the reconstruction of the connectivity of chimpanzees that were scanned, I mean, in the United States in the team of Will Hopkins, an expert and preservation of chimpanzees and bonobos. And so just to say that, I mean, ultra high field and extreme high field can be used for any kind of translational projects. And you can even go a step further. Let's assume you want to look at individual neurons. And here, this is an example of the Aplysia californica shell that you can find, for instance, close to Bordeaux in France. And my colleague, Louisa Siobanou, has developed what she calls microscopic MRI. She's using, I mean, manganese to enhance the MRI signal and be able to individually label the neurons of the buccal ganglia of the Aplysia californica. And so on the picture that is located in the center of the slide, you can clearly see that each brighting spot corresponds to an individual neuron. And of course, because you are using the buccal ganglia, you can do functional imaging. Depending on the fact that you're feeding the Aplysia before the acquisition or not, then you can observe the uptake of the manganese within a single neuron. So here you have the control with an Aplysia californica that was not fed, and you can do exactly the same with activation. So the Aplysia californica was fed before the acquisition, and you can see that the cytoplasm has an enhanced signal showing that you're able now, using extreme high field at 17 Tesla, to do functional imaging at the level of individual neurons. So everything, I mean, is, for more information, you can read the recent book that was published by Louisa Siobanou. And last, ultra high field and extreme high field allows you to develop molecular imaging, but of course it will require to use novel MRI contrast agents. And this work was performed by our colleagues at NeuroSpin, where they are developing novel strategies of molecular imaging using active contrast agents. And in that case, what they are using some RGD magnetosome that were naturally produced by genetically modified magnetotactic bacteria. And so those bacteria include some iron oxide that is paramagnetic, which enhance the MRI signals. So here, for instance, this is a tumor that was developed in a mice, and you can clearly see that when you inject that new functionalized contrast agent, so it was functionalized to target brain tumors, you can see an uptake of the signal that helps you clearly delineating the brain tumor. And if you combine that system with, for instance, focused ultrasounds, then you can develop new theranostic approaches where you'll be able to deliver specific either contrast or theranostic agents to specifically inject or deliver your contrast agent or your drug directly in the brain tumor. So this is an example of the system that was developed in combination with the image-guided therapy company, where people could clearly draw a C within the brain of a rat, and they could control the opening of the brain-blood barrier to deliver the contrast agents. And for instance, when you're doing that, you can clearly see that if you inject exactly, you can draw your region of interest in the middle of the brain at the place of the tumors, and you can see that contrast agents specifically target those structures. So of course, the CEA is still playing an important role in the development of ultra-high field systems. And may I mention that an 11.70 MRI system equipped with a large bore was recently installed for clinical applications. And it will allow to develop molecular imaging in humans, but not only molecular imaging, because, I mean, you're using ultra-high field, you can take benefit from alternative nuclei that can be explored. And this is now the time to let my colleague Fawzi to discuss about heteronuclear and MR imaging and spectroscopy.
Okay, so thank you, Cyril, for the introduction. So I will indeed talk to you about ultra-high field MRI imaging and spectroscopy using non-proton nuclei, and especially the application to investigate metabolism. To begin with, I will shortly present you the method that we are developing and validating here at NeuroSpin and elsewhere in the ultra-high field community. And I will give you some example to illustrate the potential of these tools for translational research in neurology, psychiatry, or oncology. So first of all, you have understood that you can do extremely great things by imaging water, but beyond this signal of water, and especially from the hydrogen of water, we can have access using MRI to a plethora of less intense NMR signals that can be detected using these ultra-high field magnetic fields. And those signals originate from either metabolites, which are small molecules in our brain, such as amino acids, neurotransmitters, energy substrate, or even from electrolyte, such as sodium ion and such. Some of the signals can be obtained from a non-radioactive stable isotope of nuclei, which are not necessarily visible using NMR, but these nuclei, for example, carbon-13 or deuterium, can be used to label energy substrate or amino acid to follow their metabolism and evaluate the intensity, the fluxes of these nuclei through these metabolic fluxes. Also, one can use nuclei that are completely absent in vivo, such as fluorine-19 or even lithium, which is only present in the tissue if somebody take it as a drug, to study pharmacology and molecular imaging. And so maybe I will move now to make a quick detour into the realm of physics, just to make you understand what is NMR spectroscopy. So as you know, probably MRI and spectroscopy rely on the nuclear magnetic resonance phenomenon, NMR. And this phenomenon states that when a nucleus with a spin, which is some kind of angular momentum, is subjected to an intense magnetic field, this spin will orient itself along this magnetic field, and the nucleus will rotate or precess at a frequency called the Larmor frequency. Which is proportional to the intensity of its magnetic field. However, all nuclei do not precess at the same frequency due to the influence of their surrounding electrons. This leads to a shift in frequency that we call chemical shift, as this shift depends on the molecular environment of the atom. By exciting those nuclei and collecting their NMR signal, we can then analyze the spectral component of this signal, and in the case of proton NMR spectroscopy, get rid of the overwhelming signal of water to obtain a signature of the brain biochemistry, with, as you can see on the left, with very different peaks that are specific to the main metabolites in the brain. The first conventional spectroscopy approach is to acquire this spectrum from a voxel, which is a box encompassing, for example, here in red, a region of interest in the brain. This way, we can estimate the concentration of up to 20 metabolites in the brain at a high magnetic field, such as 17 Tesla, using proton spectroscopy, or about 7 phosphorylated metabolites using phosphorus spectroscopy. This metabolite could be ATP, phosphocreatine, and such. A second approach, which is more and more popular, especially in the ultra-high-field community, is spectroscopic imaging or chemical shift imaging. And for this, we have to acquire a matrix of spectra to establish metabolic map, either in 2D or 3D, depending on the time and the sensitivities that we can have access to. And for most of the situation, we would look at the most important metabolites, such as inositol or N-acetyl aspartate. And second, then we have the non-proton imaging approach, or X-nuclei MRI approach. So for this nuclei, we have to be very careful to use dedicated sequence, mostly for the nuclei which possess spins larger than 1/2. For this nuclei, there is a rapid T2D signal decay due to their strong interaction with biological medium, and so their signals are short-lived. Therefore, it's important to use dedicated imaging techniques such as ultra-short echo time imaging sequence combined to optimize spatial encoding scheme to maximize the signal and minimize the T2 star weighting. Also, we can, as an alternative to chemical shift imaging for the nuclei such as phosphorus, for which there is a sparse metabolic content, we can use multi-Dixon or frequency selective approaches that can be used to do 3D imaging in a short time. And a third example of approach for metabolic imaging using MRI is CEST. So CEST stands for chemical exchange saturation transfer. CEST allows enhanced yet indirect detection of metabolites with labile protons, such as those found on amine, amide, or hydroxyl moieties. And thanks to their exchange with the proton of water, it is possible to generate a signal reduction of the water peak when irradiating one of those specific exchangeable protons. CEST MRI is particularly interesting for the imaging of metabolites such as glucose or glutamate. And maybe finally, a quick word about dynamic spectroscopy approach or imaging approach. Here, the idea is to administer energy substrate labeled using carbon-13, as I said before, or deuterium. For example, glucose or acetate or lactate, and to track their accumulation into several metabolic pools, such as glutamate or glutamine, which are easily detectable using spectroscopy, and to evaluate the rate of energy synthesis pathways such as glycolysis or the carboxylic acid cycle in neurons and or glial cells. So, reminder of a very important point that already Cyril made before. Spectroscopy, and especially non-proton MRI, suffer from intrinsically lower sensitivity and in vivo concentration than compared to the imaging of water. Usually we have around 80,000 times less signal and even less than that. It's so very important to use ultra-high field MR scanner to alleviate some of their limitation in terms of spatial and temporal resolution and to improve their sensitivity almost in a quadratic manner. So MR-based metabolic approach also benefit from ultra-high field from an intrinsically improved spectral resolution, which helps in discriminating between the resolution of those neurometabolites. Additionally, CES techniques benefit from the fact that T1 relaxation times are usually longer at more intense field, improving the efficiency of the saturation transfer as illustrated by those in vivo spectra. Here, for example, for glucose, lactate, and glutamate. And we can see between those two sets of Z spectra, which are the CES contrast, depending on the frequency of saturation, that we can achieve a much better discrimination between those metabolites and also higher contrast intensity. So maybe now we'll move on to some example, and I will try to give you a taste of what those methods can be used for. So while the weight of the brain accounts for only 2 to 3% of the body weight, its energy consumption is about 20% of the overall energy expenditure of the body. Most of its energy is consumed by neurons as oxidized glucose to fuel neurotransmission, the rest being used for what we call housekeeping. It is believed that in neurodegenerative disease, the frailty of some neurons leads them for diverse molecular, genetic, or environmental reasons to a vicious circle of events that accelerate their demise from energy deficit to accumulated oxidative stress to glial activation, failed homeostasis, and eventually neuronal death. And some of these events can be investigated directly using NMR spectroscopy and non-proton imaging. In fact, most of these events can already be observed in healthy elderly as a consequence of natural aging, which leads inevitably to accumulated oxidative stress and reduced energy synthesis capability. And here, for example, in this study, we've seen in elderly aged around 70 years old and 80 years old that, for example, the metabolism in astroglial cells was increased by up to 58%, and the neuronal metabolism was decreased by 28%. In addition, the neurotransmission itself, glutamatergic neurotransmission, was decreased by 24%, which is consistent with the fact that whatever happens, the brain function and vitality decrease with aging. And the idea is to be able to detect those changes in younger population affected by those metabolic neurodegenerative disease. So that's indeed the case, for example, even in kids with neurodegenerative disease. In this case, we were looking at kids suffering from metachromatic leukodystrophy, a genetic disease that affects nerve development and eventually leads to this kind of vicious circle in neurons. Using NMR spectroscopic imaging, we observe decreased level of metabolites, mostly compartmentalized in neurons, for example, N-acetyl aspartate or glutamate. And we also observed in parallel increased level of metabolites concentrated in glial cells, such as myoinositol or choline. And these are both signs of, on one side, neuroinflammation, and on the other side, neuronal suffering, especially the accumulation of lactate, which is a consequence of impaired metabolism, not enough oxygen, not enough eventually mitochondrial activity. So we can also look at neurodegeneration using sodium imaging. Sodium imaging is interesting because we have quite a lot of sodium, in fact, especially in the extracellular compartment. We have around 140 millimolar, which is the second most concentrated nuclei behind the hydrogen nuclei in water. And so we can try to look at changes in sodium concentration in neurons because sodium concentration is usually strongly regulated through the activity of NAK ATP pumps to limit the concentration of sodium inside the neurons and the different cells to 50 millimolar. But when there is a deficit in energy, this concentration inside the cells is usually going to increase because the functioning of the pump is too costly. And so what we can try to do is to look for the early sign of neurodegeneration through increase of the sodium content in the brain. And that was been done, for example, for in Alzheimer's patient, but also in patients suffering from multiple sclerosis in the left, or Huntington disease in the right, in Germany and in the US. And we can also use spectroscopy and non-proton imaging to look at psychiatric afflictions. And in those cases, what we will mostly do is whether look for evidence for imbalanced neurotransmission, or for to look for the response to treatment on those metabolism and metabolite levels. And so I'll give you two examples. For one example first, in rodents. So we conducted a study to look at the effect of chronic stress in rats. We have colleagues in Braga, in Portugal. And what we've seen is that GABA and glutamate levels were increased in the hippocampus of the rats which were more susceptible to stress. And those changes were not easily reversible, even after the stress was stopped. And that's very similar to what is observed in depressed patients or patients suffering from post-traumatic disorder, which also have GABA and glutamate level, which are often altered. And eventually, those neurotransmitter levels can be adjusted or counterbalanced using drugs such as lithium salts, for example, in bipolar patients. And that's why we are also working on lithium imaging, because MRI is probably the only way to image lithium non-invasively in the brain of those patients. And so we are conducting studies to image lithium concentration in the brain and to look for those metabolite response to lithium, whether in the area where the lithium concentration is high or low, and to check if these distribution and metabolic changes can match with the effectiveness, the efficiency of the treatment in those patients. Finally, those techniques can be used to look at the metabolic activity in the brain, especially in brain tumors. And for example, CEST imaging can be used to image glioblastoma and help in grading those tumors. That was, for example, the case for a study from EO. We used a panel of CEST approach from the APT to the NOE approach to look for the semi-mobile macromolecular and peptidic content in those tumors, which is very different, and they can also look for pH imaging. In a more specific manner, Miriam Shomei can also has used hypopolarized carbon-13 spectroscopic imaging to discriminate enzymatic activity, in particular here, to discriminate between genetic variants for the isocitrate dehydrogenase enzyme in a mouse model of glioblastoma. And it's of interest because some treatments do not show any efficiency depending on this kind of variants. So it's important to be able to have this kind of information before starting a treatment in humans, for example, and avoid a failed and useless treatment. And maybe a third example, in this situation, we are using deuterated spectroscopy and spectroscopic imaging It's a work from Ank de Feiter in Yale University, where I did my postdoc. And what they did is they are injecting deuterated glucose as an alternative to FDG PET to look at the fronting activity of the tumor in the brain of the patient. And the main advantage of this approach is that they can look at both the aerobic and the anaerobic glycolysis through the accumulation of labeled lactate or the accumulation of labeled glutamate. And so, in conclusion, I hope that despite the short length of this lecture and the relative technicality of these metabolic imaging approaches, I've piqued your interest and convinced you that in vivo NMR spectroscopy and non-proton MRI are promising tools to investigate brain metabolism and pharmacology at unprecedented resolutions. Thank you.
Thank you so much for your presentations, both Cyril and Fawzi. We have a few questions from the audience. The first one is asking about ultra-high field MRI is a very powerful platform for experimental medicine, but it's only available in several expert centers. What are the current bottlenecks to transfer the findings to conventional 1.5 or 3 Tesla systems to have bigger clinical impact?
Well, first, 7 Tesla imaging is really democratizing, actually, because now there are around 70 such systems in the world, and because the manufacturers get the FDA approval, those systems turns to be now real clinical devices. So they will probably be spread out of the clinical hospitals, which was not the case before. And second, how to translationally take benefit from the results obtained at 7 Tesla to enhance results at 3T and 1.5T. The thing is that because at higher field you can better understand the structure and the functions, then you can put some prior knowledge in the process or image processing pipeline that you're using at lower fields to be able, even to get some functional or structural information that you cannot clearly see at lower field, but because of those priors, you can get some proxy to the same kind of information. May I say, for instance, that if you're doing a functional imaging at 70, your resolution will be some millimeter, and it will allow you to, for instance, to distinguish if an activity occurs in one or another layer of the cortex with respect to a given stimuli. Then you can use that information and to do an acquisition at lower field, observe the activation at a lower resolution, but because you know that it occurred in such or such layer, then you can project back your information to the adequate layer. Maybe I can let also Fawzi comment on this.
Okay, so I agree with Cyril that for some of those techniques, the addition of prior knowledge can help narrow down the relevant information and eventually be able to achieve some of this measurement. But for some of them, it will be complicated to assume too much about those information or the distribution, for example, of such and such metabolic information. And so I don't know if it's going to be easy to translate most of those techniques to lower field strengths like 3 Tesla. And another obstacle is that, especially for non-proton MRI, you need to have a dedicated hardware, which means that you need to have a dedicated transmission to excite the spins at the resonance frequency, which is not the same as for the hydrogen nuclei. And also you need the reception coils as well to detect efficiently those signals. So it's one other challenge that is on our way if we want to translate those techniques to 3 Tesla. Maybe just a quick word about some of those approaches, metabolic approaches, can be used using hyperpolarized nuclei. And in that situation, the hyperpolarization is not depending on the field strengths at which you do the observation, but depends on the availability of hyperpolarized substrates or something like that. And that's a different matter. It's probably less costly, but it's very limited to fast, easy to access enzymatic activities.
Okay, great. Thank you very much for your answers. There's another question about the presentation highlighted the theoretical aspects of ultra-high field MRI, MRS, and some diagnostic applications. Can you mention an example where it supported drug discovery and/or target validation? If not, in which indication or drug class do you expect these techniques to have a major impact?
Okay, so first question, if I understood correctly, is the question is, are these techniques useful for drug discovery, or have they already been used for drug discovery? Well, I don't really know, because whatever happens for drug discovery is not necessarily public in most laboratories, so I cannot rule that out. But in any case, it's also very interesting for better understanding what the drugs are working. In fact, most of the drug discovery are not really looking for most industry is not really interested in understanding how the drug is working, and that's something that we can try to do with these techniques to understand how it works. And also, it's interesting to know how it works in combination to eventually other drugs or other conditions that were not necessarily the first one that were being investigated.
May I add that, I mean, a pharmaceutical MRI, which is the typical name for the study of drugs using MRI, I mean, is very useful to see, I mean, the status of the brain after taking a drug. So typically you're doing a rest extended functional MRI before taking a drug to see your current status without a drug. And then you do the same after taking the drugs, and you see how the drug has modified your default mode network. So it gives you some insight about the real effects, functional effects of your drug on your brain functioning.
Yeah, just to go back to that, in the process of drug development, what's really important is it's a good clinical effect, okay, at the end of the day. But eventually, for very specific question, for example, for dose adjustment, so to fit the dose of the drug, if there's not a lot of difference between two doses, it could be interesting to have these approaches to discriminate between the amplitude of the, for example, neurotransmission, the changes on the neurotransmitter levels according to the dose, and see if there's a correlation between or difference between the metabolic changes and the efficacy of the drugs.
Thank you very much. We have now come to the end of this webinar. I would like to thank you all for attending today's webinar, with a special thanks to our lecturers for their excellent and very informative presentations. Should you still have any further questions to one of the speakers, I welcome you to submit those to us by email, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our website within 2 weeks. We hope to see you all online again soon. Thank you again and have a nice day.